Welcome, fellow science nerds, to part 2 of microevolution or population genetics. So let's go back to our previous example. In our previous example, when we discussed the incomplete dominance in flower color, we calculated the dominant allele, or rather the incompletely dominant allele, to be the frequency to be 0.8, whereas the frequency of the recessive allele was 0.2, if you remember. So using this, we can actually calculate the frequency of genotypes using the multiplication rule. If you remember in genetics, we use the multiplication rule to calculate probabilities of having offspring with a certain genotype. So the multiplication rule is a rule of probability, so it can be applied to this instance as well. So what is the probability of getting a homozygous dominant individual? Well, if the allele frequency is 0.8, then the probability, and the homozygous dominant individual needs to have both dominant alleles, then the probability of getting a homozygous individual is 0.64, because it's 0.8 times 0.8. Okay, so the frequency with which we expect to see a homozygous dominant individuals or red flowers is p squared, because we multiplied the frequency of the dominant allele. P times p is p squared, or 0.8 in this case, or 0.64. Similarly, the frequency with which we expect to see white flowers is q squared, because we multiplied the frequency of the recessive alleles. Q times q is q squared, or 0.04 in this case. Now, how do we get a heterozygous individual? Remember, we can get it two ways. One parent can donate the dominant and the other one the recessive, or vice versa, recessive and dominant. So it is 2 times 0.8 times 0.2, or 0.32. So the frequency of the genotypes can be confirmed using a Punnett square. So if P and Q represents the relative frequency of the only two possible alleles in a population, then in a particular locus, then we can say that P squared plus 2PQ plus Q squared equals 1. Why? Because the frequencies of the dominant, homozygous dominant individuals, the frequency of the heterozygous individuals, and the frequency of the homozygous recessive individuals must add up to 1, right? We only have 2 alleles at this particular locus. Now, P squared and Q squared represent the frequencies of the homozygous genotypes, whereas 2PQ represents the frequency of the heterozygous genotypes. And again, this has to do with the probability rules, the multiplication. So natural selection, genetic drift, and gene flow can alter allele frequencies in a population. They can cause a population not to be in Hardy-Weinberg equilibrium. So let's explore them a little bit. Natural selection causes adaptive evolution. That means that the population will change in such a way that it is now a better fit. Its individuals are better fit to their particular environment. So traits that enhance survival and reproduction increase in frequency over time. That's called adaptive evolution. Now, genetic drift is a process where the allele frequencies will change unpredictably. They will change from one generation to the next. It's simply chance. So this happens all the time, but the smaller the sample is, the more likely that chance alone will cause a large change from a predicted result, right? I mean, this again, it's a matter of probability, right? So the smaller the sample size, the more likely it is that random chance will cause a drastic change from what you expect. So genetic drift actually usually reduces genetic variation through loss of alleles, especially in small populations. And so here is an illustration of what genetic drift looks like. It's basically randomness. Genetic drift is randomness, but this randomness has consequences in a small population. The randomness is present in all populations, but if the population is large enough, that randomness gets lost, it gets diluted. But in a small population, it can have dire consequences. So let's look at this example. We have here 10 flowers. Again, we're going back to our incomplete dominant allele, the pink, red, and white flowers. And we have 10 flowers. Some of them are red, some of them are pink, some of them are white, right? And we calculate the allele frequencies is 0.7 for the capital R, the red allele, and 0.3 for the white allele. So, but we have only 10 plants, okay? So by pure chance, only 5 of these plants leave offspring. The other 5 are, let's say, not pollinated or not pollinated successfully. It happens, okay? So only 5 of these plants leave offspring. So now generation 2 and the 5 that leave offspring are highlighted in yellow, you can see. So in generation 2, now the allele frequencies looks very different. Now we have 50-50, so P 0.5 and Q 0.5. Why? Because only 5 of the 10 flowers left offspring. It's just pure chance. Which ones are going to leave offspring? Now, out of this new generation, generation 2, which is again only 10 plants, only 2 of the plants actually successfully reproduce. And by chance, those 2 plants are the red plants, right? So the first time we had some red plants, we had some pink, we had some, even one white reproduce. But the second time around, by pure chance, only the red flowers reproduced. So the third generation is going to look completely different than the original population. Now the recessive allele is completely lost from that population. In that population, the incompletely dominant red allele has become fixed. It is the only allele present in the population. All the individuals are homozygous dominant. Now, generation 3 population has a lot less variation. Because at this particular gene, there is no variation. And this is all because of genetic drift, because of randomness. Now, another thing that could happen is the so-called founder effect. The founder effect occurs when a few individuals from a population become isolated. So if you have a large population and a few individuals become isolated from that larger population, you now have a small population really. Allele frequencies in this small founder population can be very different from those in a larger parent population due to chance. And another phenomenon is the so-called bottleneck effect. Now, the bottleneck effect can result when you have a drastic reduction, a significant reduction in population size due to a sudden environmental change. That environmental change could be a climate change, could be over hunting or harvesting by humans, habitat destruction. It could be many different causes that would lead to a drastic reduction in the population size of a particular species. And by chance, the resulting gene pool may no longer be reflective of the original population's gene pool. If the population remains small, it may be further affected by genetic drift. So when you combine the bottleneck effect with genetic drift, you are in danger of this population actually disappearing. So bottlenecking of the wild population of the Florida panther, as shown here, is a subspecies, has caused this subspecies to be on the brink of extinction. And here's another illustration of how the bottleneck works. And it's called the bottleneck because this particular model shows you how it works. So you have a large population, original population that is very diverse. You see you have white, yellow, blue marbles. Let's call them marbles. So you have 3 different alleles. There's a varying frequencies of these alleles within the population. Now something happens. And now the only surviving population it's like the population is very small in size and there's very little diversity, very little variation. There's only mostly blue and there's like 1 white marble. So this is referred to as the bottleneck effect. This population has low diversity, low variation, and on top of it is also very small. So genetic drift can contribute to further endangering this population of losing all variation. And a population that has low variation is more susceptible to changes in the environment, and because there's so few variation in the population, that population actually is in danger of going extinct. So let's summarize the effects of a genetic drift. Genetic drift is significant. Genetic drift is always present, but it is significant only in small populations. And genetic drift can cause allele frequencies to change at random, because it's just randomness, it's chance. It can lead to loss of genetic variation within population, and genetic drift can cause harmful alleles to become fixed in the population. That means they're the only alleles in that population. So genetic drift can be actually quite dangerous to a population. So let's talk about gene flow. What is gene flow? Well, gene flow is the movement of alleles. So you can get new alleles in the population, or you can lose alleles. And that is caused by migration. So alleles can be transferred through the movement of fertile individuals or gametes. So if it's animals, animals migrate. They migrate into or out of populations. If it's plants, it is the gametes that migrate. So pollen is carried by pollinators from, pollen can be carried from pollinators from one population to a different population of the same, of plants. So that could cause alleles to flow in and out of a population. Now, gene flow could have varying effects. It depends. It could reduce genetic variation among populations over time, but it could also increase it if new alleles are flowing into a population. But most likely it will reduce genetic variation. Gene flow can also affect the adaptation to local environments. So you have a certain type of alleles present in a population. That population, remember, those are individuals of the same species, but they're adapted to a local environment. A population of the same species that live in a different environment may have different, will have different alleles, because it may have different adaptation to a different environment. So the flow of alleles in and out of population can actually affect the adaptations to local environments. It could improve them or the other way around, but it is an important factor to consider when we talk about allele frequencies and evolution of populations. Okay. Oh, and we are slightly stuck. So in summary, when we consider gene flow, when we consider genetic drift, when we consider natural selection, these are all factors that are constantly at play in population. We can understand why a Hardy-Weinberg equilibrium is a very theoretical thing, and it's rarely seen in nature. Okay, we're going to talk more about Hardy-Weinberg next time. So for now, this is goodbye from me. Have a great day, guys.